It's special. It's special today.
Okay, I'm gonna explore.
Okay.
Is it working?
So, I think it's working.
Do you want me to like check on my laptop?
Yeah, could I actually give you a link and can you
Yeah.
actually, well, let's see, we could even just follow it. See if I still have pasted the correct link. No, what the fuck? I hate technology sometimes. No, really. Okay, here is this link. If I follow it. If you go to YouTube, search Norian.
Alright, so you see this dip scan. I think you only need this time. I don't need this heavy one.
Here you go.
Thank you.
You're welcome.
How do I advance this, Sam?
Oh, you gotta turn it on. There you go. Is that amount of weight okay?
Is it the thesis
Yeah.
real one?
Yeah.
Yeah, it's working.
Okay. You can see me right now?
Yeah.
How's that? That's
Alright, well I
right?
can't see you, but
You can see
I
Is
the
that right?
see the
Is it at the bottom
presentation.
okay? So you gotta turn it on.
Where's the on?
On is here.
Oh, I see. Okay.
In fact, the one thing I should make sure of is I'm pretty sure these are pretty new. I think I changed them when I went to Michigan in October.
Okay.
And
Great.
then forward, backward.
Okay.
Okay. And you don't need to touch anything there. I left this here so you don't misplace it.
Perfect. Thank you.
Okay, and when you push that thing around, if you have a
Back.
yeah, but you also can, if you really want to. I think you were.
Oh,
Okay.
what am I doing?
Okay.
Hi.
Great. I think the amount of weight I put up
Hello?
there is just right. It's pulling it down to allow everything to.
Dad?
it's kind of delayed. Like
That's
this-
okay.
Okay.
Are you gonna be managing the video?
Or I was just checking if it was working,
that
but
it could work.
Okay, it all works?
yeah.
Okay.
Shit. Okay, let me call my dad back. Sonia, could you hear?
Let me. I'll go
Sound?
outside and check.
Okay.
If I I can like
Oh,
hear you.
that's good. Margie. Okay.
Oh, I see. It's changed.
Oh, good. Yeah.
It's been like 6 years.
What? No, I've
They
met
never
her.
met?
Yeah, I met her.
No.
Oh,
your
my
dad,
dad. Yeah, no, they haven't met either of my parents actually.
Oh, you're so smart.
Oh, yeah.
Good luck.
Okay.
Alright.
Oh my gosh. Hi. Yeah, you can.
Yeah.
Okay. Oh my gosh. Hi.
Hi.
Oh, it's weird being this tall.
I know.
Like, I'm not short to begin with, and then I'm like, let me add
How
4
high are
inches.
your heels?
Well, they're wedges, so they're
Oh, wow.
comfy, but
They're high.
they're tall, yeah. But they're my favorites. I can walk around in these all day without, like,
Yeah.
practicing.
Nice. So excited.
It's super gnarly, guys. It's crazy. Yeah, this is okay. Hi.
Hi, have fun.
Thank you.
Thank you.
Oh, don't do like that.
Wherever you want.
Wherever you want.
Up in the front, in the back, wherever you want.
Just grab the spot.
I guess I'll camp out.
Everybody, would we like to start making our way in here? Marcy?
Hi.
Hi.
How you feeling?
Pretty good. How are you?
I'm good.
Yeah. Did you get to read that thesis yet?
I've been paging through it,
Yeah.
so I've read enough of it. Don't worry, it's really good.
Thank you.
Bobby Nick. Hi.
Thank you for
Oh,
coming.
Is your
Yeah, she's running a little late. My brother is too.
Missed a lot of traffic.
But like,
Are you recording this for me?
yeah. So don't say anything you don't want to be forever eternalized sane.
I tried. I know.
Let's see if that works. Yeah, it does work. Success. There's power.
do you have an ETA for your mom and your brother? I can't believe it.
My brother said 10:10.
Shouldn't be doing this at all. You know how
long your class is, by the way? Is it 60 or is it 45?
Maybe around Monday
It's
morning.
gonna be probably closer to 60. Yeah.
It should be around
I was gonna say,
Sunday.
they even have the option of waiting a couple minutes to make sure they arrive.
Yeah, I'm asking her right now how late she thinks.
Hopefully they're parking right now, or?
Yeah.
Ralph's? Yeah, it's Ralph's. It doesn't have to be from a French bakery or anything. Time. Fantasizing about
Hey,
what?
where are you?
Yeah, but I wish I could say, the problem is with shuffling kids around. I'm looking to be at 10.
Yeah, okay, you're gonna be a while. Like, you're looking at a half hour. So we're probably gonna get started, but maybe we'll wait a few minutes to speak. So it'll be a little bit Jake said 10:10. Daddy's here. Okay, I gotta go. Bye.
You can introduce
What's the story, Should we start?
If we could wait 5-10 minutes, they'll still probably be a little late, but that would be ideal.
So we'll wait 5 more minutes. Is that all right?
How about 5?
My family.
Do they know how to get to this room though?
Yeah, I gave them. Did they work okay? Yeah.
I went to the lobby and asked.
And do they walk you over there or do they just direct you down?
Actually, I made my own detour and I found my way there.
Excellent. Alright.
Should we prop the door open or something?
I'll go in, but I don't need to know.
Oh, yeah. Super.
Sorry. Okay.
Depends on who you talk to.
Do have to introduce you.
Do you want to start introducing?
They tend to be a little tougher. I'll wait a couple minutes.
Okay.
So you might want to.
Is your video on?
Uh-huh.
Okay, that's really fair.
You might want to have a seat.
In the cabin?
Yeah, I'd say wherever you want.
Okay.
I didn't make the case that there should be.
Yeah. Yeah, it's good. It's good. I was trying to see if there's a code up there in colors.
Oh.
So if you look at the cyan, it's kin hot hot. I don't know what that means. Mo east ants.
I didn't realize that. If you put the blues next to the reds, you could have arranged the colors in such a way that parts of the display would have come out. complementary colors right next to one another, you can get effects like that. There's an Austrian author, William still alive, his name and he started doing films after the Second World War. So they were very much influenced by the war. A lot of them were buildings that were usually exploded. But he had a huge number of tapestries, big collection of his art at the Modern Museum in Paris, where we happened to be in Paris. I got halfway through this exhibit before I realized that he was all over the place doing these things with complementary colors. And all of a sudden I saw it. And once you see it, they were wearing sort of a dress, and you start seeing it everywhere. We had to go back to the beginning. I think we'll start now, at least with the introduction. We're waiting, have been waiting for Nori's family, who will come. But, they missed the very beginning.
Do they know about the traffic in Westwood?
My dad's here.
Okay.
All right. So this is a very happy day in anyone's life, getting finally through with the PhD.
Well, now hang on a second. Sorry.
I actually read
It says candidate.
I actually read the thesis. Did you?
Yes.
Let me introduce Nori. Nori grew up in Victorville in the high desert, or at least bordering the high desert. I don't know whether you consider Victorville actually part of the high desert.
Yes, very much in the high desert.
You do. Yes. I've been there many times because my father lived in the high desert for many years. And then she then came to UCLA as an undergraduate. And after finishing her 4 years and not really knowing what direction she wanted to go, but being interested in research, she decided to do the master's program at UCLA. And she worked in Patty Phelps's laboratory and produced a nice first authored paper and finished her master's, and in the process got the teaching award for TAs, which is extremely competitive, and it reflects her abilities as a teacher, and you will undoubtedly see some evidence of that in her seminar today. When she was doing her master's, she took 173, which is the course that I taught then with Mark Fry and Peter Nairns and Alan Grinnell at some point, depending upon the schedule, and which Sam is now participating in. This is the course, the upper division course in sensory physiology that the Department of the IBP, what used to be physiological science, offers to undergraduates. And Nori got very interested in phototransduction and came to me and said, "Can I see an experiment?" And so I said, "Sure," and we arranged for her to sit in on an experiment with Mike Woodruff. And even on the very first day, she started playing with the manipulator, and by the second time she looked at the experiment, she was actually doing recordings. And then she started doing the dissection. Needless to say, she learned very quickly. And so with my encouragement, in fact, almost my insistence, she applied to the PhD program and began working initially in my laboratory under Mike's supervision, and then moving to Sam's laboratory to exploit the techniques that Sam uses to record from retinal slices, and you're going to see lots of evidence of that today. So it's with great pleasure that I introduce Nori, who's going to, I won't read the title of the seminar because you can all read it yourself, and she can tell you about not everything that she did, okay? There are 3 more papers that she's not even going to talk about, but this is the, I think, what we would consider the heart of the research that she did. Nori,
Hi everybody, thank you for being with you the work that I've spent a few years on at this point. So today I want to show you how I've unlocked mouse cone photoreceptors, and we're going to look at how their phototransduction and biophysical characteristics play into the detection of light. So, we are vertebrates, we are mammals, and we have eyes that look like this. And so, if we are going to collect light information and do light detection, light first has to enter through this translucent window of the cornea, pass through an adjustable pupil aperture, and then be transmitted through a lens, which will focus the light on the back of the eye, where the light can then be absorbed by the neural retina. For light to be absorbed through the retina, it has to pass through these three major layers of cell stomata before it's absorbed in kind of this orange layer, which consists of the rod and cone photoreceptors. After light is absorbed, it's passed down to these bipolar cells in green, where parallel processing occurs. And then finally the information is passed to the ganglion cell layer. These cells are spiky neurons, and they have long axons which are going to transmit the electrical signals of light to cerebral structures. So, as vertebrates, we have evolved what's called a duplex retina. And this means that we have two types of photoreceptors, rods and cones, that mediate the detection of light we use for image formation. Now, rods have been highly specialized to operate in dim or scotopic conditions, and they can reliably signal single photons of light. Cones, on the other hand, have been optimized for bright light or ambient light conditions, and in species that express multiple subtypes of cones, the cones mediate color vision. There is kind of an in-between mesopic region where both rod and cone inputs are going to mediate light detection. Yep. Okay. So in a general sense, we have one cell, or a group of single cells, that somehow need to detect this electromagnetic stimulation from the environment and somehow transform that into an electrical signal that the brain is going to be able to interpret. And within this single cell, there's multiple events that are going to first detect and shape the output of this signal. So the first one is actual light detection and phototransduction. This is going to happen in the outer segment. And the end result of phototransduction is some sort of current change. We're going to close channels in the outer segment. It's going to change the total amount of current that the cell is passing. Now, this is Ohm's law. So what this means is that if we change current, there's going to be some sort of proportional change in voltage. So just through phototransduction, we're going to create some sort of electrical response to light. Now, that happens in the outer segment. Further down in the inner segment, we actually have voltage-sensitive channels. And these channels allow different types of ions to pass, and they're going to further shape the photoresponse. Finally, in order for this cell to signal to the brain, to downstream circuitry, it has to be able to actually transmit this signal. So they have synapses down in the cone. It has this large pedicle structure, and kind of like typical signal transmission, they have voltage-gated calcium channels at their synapses that are going to mediate the actual release of glutamate. Oops. Okay, so why do we care about the cones? Why not go to the very output of the retina and say, well, what is the electrical output here? Why do we care what the cones are doing and how they're shaping the signal? Well, there have been studies that have looked at different ganglion types and have correlated their noise in disparate types, and they're able to track this noise to the cone photoreceptors themselves. Another example of why cones are important is if you look at the theory guiding behavioral color discrimination thresholds. Current theory in the color vision field is that these discrimination thresholds are set by the noise in the cones themselves. So here you're having a behavioral output that is being defined at the first step by the initial detecting cell itself. So it's important to understand what the cell is doing and how it's actually encoding these signals and how the system is limited by the way that these signals are encoded. So that's great. Why don't we study cones? Why don't we study all the cones? Well, in mammalian species, cones really only represent about 5% of the photoreceptor population. So this has made isolation of single cells for electrophysiological or biochemical experiments very difficult. As a result, much of what is known about photoreceptor physiology has been carried out in the rod photoreceptors. So just so you have an idea, these two images are mouse retina in two different orientations. This one on your right is a flat-mounted mouse retina, and these dark spots are the cones, and the rest of them are rods. The colored image to the left is a slice going through the retina, and the pink cells that you can see in here are again the cones. Cool. Okay, so they're a sparse cell type. Well, typically today you can go into the lab and you can label sparse cell types with some sort of reporter. And you can go in and you can shine a light and you can find these cells. Well, that's a problem in our experiments because our cells are sensitive to light. And so if we want to understand the initial conditions of our experiment, we need to be able to control how much visible light is shined on these cells. So we can't use a reporter system that uses light to isolate or locate these cells. So again, just to emphasize, all my experiments are carried out in the dark under infrared illumination. Okay, so we still need to talk about how these cells are going to detect light. So I'm going to give you a little bit of background first on the molecular processing governing detection and the initial signal generation. And then we're going to switch and talk about shaping the electrophotoresponse with downstream channels. Okay, so first let's detect light and create a photoresponse. Now, in the dark, inside a general photoreceptor, we have cyclic GMP. In the dark, cyclic GMP concentrations are quite high, and when they're high, they can bind to cyclic nucleotide gated channels, CNG channels, and in the dark, a certain amount of these channels are open, and they're passing sodium and calcium into the cell in the dark, depolarizing the cell. Now, this is a special channel, and the maximum photoresponse is actually defined by how many channels are open in the dark. Okay? Let's shine some light. Okay. So this is our symbol for light, and it's going to be absorbed and activate a photopigment. This photopigment can then interact with a heterotrimeric G protein, and it catalyzes the exchange of GDP for GTP on the alpha subunit. Alpha transducin activated can then interact with phosphodiesterase, PDE, and PDE will actually hydrolyze cyclic GMP, reducing the internal concentration and closing these channels. This is initially recorded as a reduction in inward current when we look at traces. Now, to end the photoresponse, we have to turn off PDE. So there's another complex that comes along and hydrolyzes the GTP, and phosphodiesterase stops hydrolyzing cyclic GMP. Now, if there's prolonged light exposure and the channels can remain shut for a considerable amount of time, the internal calcium that was entering actually drops to very low levels. And there are several calcium-sensitive proteins inside these cells that will mediate adaptive processes. One of these proteins is guanylyl cyclase accelerating proteins, GCAPs. And these work on the guanylyl cyclase to increase the rate that it can synthesize cyclic GMP. The other calcium-sensitive protein is recoverin, and recoverin acts so that in dark adapted conditions, it allows a longer lifetime of an activated photopigment, and after light adaptation, the life of the photopigment is much shorter due to the actions of recoverin. One last protein I want you to pay attention to that's important for this talk is, in fact, this alpha transducin subunit. This is one of the main adapter proteins or transduction proteins that's going to connect this light adaptation to channel closure. And so I'm going to show you one of the mutants that has this knocked out specifically in rods, and the rods are not able to generate an electrical response when exposed to light. Okay, so moving down further into the photoreceptor, we've kind of, now touched on the C and G channels up here in the outer segment, but there are also going to be other ionic channels that you will see today. So further down in the inner segment, there are potassium selective channels. There's a channel that is activated by hyperpolarization. There are the voltage-gated calcium channels in the synapse, and there are actually an anion channel that is activated by the calcium that enters through these synaptic channels. So you're going to see some of those as well. Okay, so everything I showed you in general at this point is in general for photoreceptors, but I did explain that these photoreceptors operate in very different conditions. So while the general components of the molecular machinery I just showed you are conserved or are there, the actual little pieces or the types of proteins that are there, the isoforms are different between the photoreceptors. Now this isn't always true. Some of the isoforms are the same, but the proteins might be expressed at different levels. And there are the proteins, GCAPs and recoverin I just showed you that are identical within these cells. So if you look at the accumulation of all of those different small changes I showed you, it leads to a difference in response. So these are just saturating flashes. The black is from a rod photoreceptor, and the red is from a pure cone response photoreceptor in this GNAT1 knockout animal. It's the transducin knockout. To generate these responses, I had to flash very different light intensities to generate them. But I have normalized them so that you can see the difference in kinetics between the waveforms. So cones have very fast, quick responses, where rods have these longer, slower responses. The sensitivities between these cells are different, where cones are almost 2 log units less sensitive than rods when you measure their entire dynamic range. But interestingly, the maximum response, so like I mentioned, the number of channels that are open in the dark, which defines the maximum response, seems to be quite similar. So in this cohort of cones, I measured their maximum responses to be almost identical. So despite the differences, there are impressive similarities as well. Okay. Oops. So even though these the rods and cones have different specializations, they're closely associated with one another, and they're going to share signals with each other. Signals are allowed to spread to neighboring photoreceptors. And this happens through these structures called gap junctions. They're electrical synapses, and they're going to be found in the synaptic regions between neighboring photoreceptors. We've known about them for a really long time, so this paper is from 1973, but we've recorded signal spread as a field since the 70s as well. So you will see some of these effects in my recordings. This is another picture still showing gap junctions. These are primate cones, I believe. Okay. So carrying on, I'm going to walk you through some of my general methods. If it changes, oops. Okay. So in general, everything I'm going to show you today happened in dark adapted retinal slices. So again, dark adapted, I'm doing the dissection, I'm doing these recordings in the dark. These retinal slices are made both from wild type and transgenic mouse strains, since I don't need any genetics to identify the cell type. Once the slices are made, I'm going to use single cell recording using whole cell patch clamp, and this can be done in different configurations, which I'll go through in 1 minute. Later you're going to see some pharmacology where I use to isolate different channel components, and there's a little bit of fluorescent imaging. Okay, so let's talk about single cell recording. In a slice, in a preparation, you'll have some sort of cell, and you can approach this cell with a glass pipette. Once you approach the cell, you'll press up against the membrane and form a high resistance seal between the membrane of the cell and the glass pipette itself. Using additional pressure, you can actually kind of pop a hole in that cell so that the internal of the cell becomes continuous with your electrode. And this electrode is then connected through circuitry to an amplifier where we can record voltages and currents. So here's Ohm's law again. And basically with this experimental technique, the experimenter can either use voltage clamp, where the experimenter chooses a voltage and experimentally holds and clamps that cell at a voltage. And when it does that, you're able to record the current changes that are occurring due to stimulation. The experimenter in another configuration can control the current inputs, and when you do this, you're actually going to record the voltage of the cell itself. So this is a huge advantage and is really mostly done with whole cell patch clamp. So other types of patch clamp, you can't really record both methods in the same way. Okay. So I've kind of broke this talk into 2 parts. So the first part, I'm going to mostly focus on the cone photoresponses. I'm going to show you that I can identify the unlabeled cones. I'll show you voltage clamp or voltage and current recordings from the same cell. I want to show you some of the rod input or the rod signal spread that you can see in the cones and how to isolate that. And then I want to show you how the cones respond to brief flashes and steady light. The second part, we'll look at inner and outer segment currents. We'll look at different ionic conductances, how those might affect unclamped light response, and then I want to look at reversing the polarity of the light response itself. So, part 1 is cone photoresponses. And first, let's find a cone. So this is the retina, and this is actually a picture of my experiment, so this is what I work with. And this is, it's a little blurry, but this is basically the photoreceptor layer. And the cones are located in the outer 3-ish rows, so I know where to look for them. The cone soma are slightly bigger, and they have a slightly different pattern that's visible under IR illumination without staining. And I can identify these cones with about 90% accuracy, unlabeled. So again, this is in IR illumination, so it's not stimulating the cone, but I will find a cone. You can kind of see the shadow of my pipette. Once I gain access, pop that little hole, I can record whatever protocol I'm recording, and during that time, a fluorescent dye will actually fill the cell, and at the completion of the recording, I can actually image the single cell that I just recorded from. Now, before I take those pictures though, I can record from the same cell responses to light flashes. So in this type of experiment, I'm giving brief flashes, approximately 5 milliseconds of increasing intensity. So you can see the response increase until a saturation point. This would be where all those C and G channels I keep mentioning are closed and current goes to 0. And this is an example of the same cell responding to the same intensity flashes, but now recording the voltage responses. Okay, so this was recorded from a wild type cone. And while this wild type current response looks like this, they don't all look like that. A lot of them will look something like this, where you have a fast response, but then before returning to baseline, it kind of floats along the slow plateau before fully returning to baseline. And pretty early on, I was pretty sure that this was the rod signals spreading into the cones. Why did I think that? Well, if you look at the time course matches or is close to the time course that a rod signal will generate at these potentials, and it also seemed to increase with intensity. I could apply a really dim background and get a preferential suppression of this rod input, so I'm saturating the rods but leaving the cone mostly intact. So this is a dim background with the cone flash on top of it. And then it was possible actually to look at genetic mutations and get these very fast monophasic pure cone responses. So this is the alpha transducin knockout in the rods. So the rods in the retina are intact. They can be bleached by light, but they produce no photoresponse. And from a single cone, you can collect the entire pure cone photoresponse. In another mutation, our transgenic mouse, connexin 36 knockout, this is the gap junction that's actually going to allow the signal spread between the rods and the cones, or the cones and the cones. And you get very similar monophasic fast responses without that slow component. So rod signal spread does occur in wild type to cones, but we can use these different genetic backgrounds to actually isolate the cone response. And I do use both of these extensively through my experiments. Okay. After collecting flash families, we can measure a variety of parameters describing these flash responses. So at the top, I'm measuring intensity versus response. So what I do, go back, is I take the amplitude of each of these flashes, typically normalize them, and plot them against the intensity of the flash. This black line data represents the sensitivity of the rods, or the dynamic range of the rods, and these red and blue traces represent pure cones. So you can see this rightward shift represents almost a twofold decrease in sensitivity between the rods and the cones. This pink trace is actually cones that are coupled to rods. They seem to saturate at similar intensities that pure cones would, but when you look at these kind of dimmer responses, they're producing larger responses to dimmer light because they're kind of boosted by that rod input. Another parameter we can measure is looking at basically the gain of the response, or how well the phototransduction cascade is activated by photons. So for each of those flashes I showed you before, you can fit them with an equation and derive a parameter that tries to describe or capture how well the transduction cascade is activated. And so it's pretty constant across most of the dynamic range in the cones. I didn't include the coupled cones in this analysis because the rod input actually confounds these analyses. So this is for pure cones. Okay. So I've shown you some of these brief flashes, but cones operate on a background, okay, or they're optimized to do that. So what happens when you shine continuous light on these cells? Well, they both hit at some point a transient saturation point, and you can see between the flashes and the steps, these are from the same exact cone, that this seems to be pretty stable. So this is when all the transduction channels are closed and current goes to 0. Well, in cones, there's a considerable amount of relaxation. Even after you have a response that's closing all the channels, a large fraction of those channels might be reopening back up. And the brightest step here is bleaching 1.5 million photopigments per second. It's insanely bright, and still the cone is showing a certain amount of relaxation here. So the question now is this a functional recovery? can we now flash another light on top of this background and reclose these channels? Or is it just some sort of other current that is helping to bring the membrane potential back down from the saturation point? Okay, so this basically says what I've been talking about. They do seem to hit a saturation point, but they quickly recover. And is that functional? So in an experiment like this, I, in the dark, first record the flash family, the brief flashes to the cell, to get an initial sensitivity or dynamic range reading. So the red trace here is just replotted down here so you can see amplitude and scale. So after recording flashes in the dark, I can apply a background, which is moderately less bright than that brightest step you saw, but nonetheless gives you an initial saturation point. After the background comes on, the background is constant, but the cell relaxes, and then I begin to give flashes on top of that background. And with these flashes, I can resuppress a lot of the photocurrent, almost basically to the saturation point here. These cells never exceed this level, right? Their amplitude never goes past this initial saturation point. And when I look at their sensitivity, this background shifted the sensitivity about 13-fold. So here is those flashes isolated here. But yes, basically after the cell is recovering, those cyclic nucleotide channels are reopening, and you're going to be able to resuppress them with background light. Okay, so I went back then and started looking at what happens to cones when you knock out some of these adaptation proteins and record them with brief or steady light presentations. If you knocked out recoverin, the flash responses weren't dramatically changed, and the photo response or the steady light responses still had quite a bit of recovery. They did seem to be a little bit less sensitive than wild type. If you knocked out GCAPs from the cones, whether or not it was with recoverin, the response time course got a lot longer, and this causes the cells to seem more sensitive to light. Another big change was the actual amount of recovery that occurred from that transient saturation point to steady state. And there was much less recovery happening when you knock out GCAPs versus recovery. So this would indicate that GCAPs are really important for the calcium dynamics that are going to set the actual steady state level in a certain light intensity. So conclusions for part one, I can very easily identify correctly unlabeled cones. You saw that the broad input can spread into the cones, and you can suppress it with a dimmer light. And we saw genetic manipulations can actually be used to isolate the cone response. Cones escape saturation even in very bright backgrounds. And that is a functional recovery. And we saw that knocking out some of these calcium-sensitive proteins that are involved in adaptations has profound effects on how the cone is going to operate in steady light conditions. Okay, I want to move on to part two now and talk about these ionic conductances, unless there are any questions. No? Okay, so just as a reminder now, we are trying to detect light. We talked about this first section, so we're going to now concentrate down here. How are we going to now affect, shape, or change, modify this initial signal that we created up here and transmit it to the rest of the retina? So again, just as a reminder, I showed you these channels before. And now for this section, I just wanted to point out that for each of these channels, there is a specific blocker or combination of blockers that are going to allow me to isolate these different current components. Yep. Okay, so I'm going to give you a little bit of background also for these experiments so you know what's going on. So we're back to our voltage clamp cell here, and in voltage clamp, we pick a V-hold, a holding potential, and in the previous experiments that I showed you, I basically did this, where I held the cell at a constant level, gave a flash of light, and recorded the response. Now for these next experiments, we're going to pick a holding potential, but now we're going to shift it to a new potential. And when we shift it to a new potential, it should elicit some sort of current change that we're going to be able to record. And now we're going to kind of dissect out some of this weird, wacky stuff that seems to be going on when we shift the holding potential. So a few things we can measure from this is we can actually measure how the current relates to the holding potential. We can look at the voltage that the response actually will change polarity, the reversal potential, and this is important because it allows us to predict the magnitude and the direction of where current will flow. And using some of the blockers and the reversal potential, we can look at permeability and specificity of these channels. So I want to start with the hyperpolarization activated channel, or IH. So this is activated by giving negative going steps, and these are the currents recorded. I can fit these currents with an equation that allows me to quantify how fast this channel is activating. And so when I start to get fits, it starts to activate, or it activates by 25 milliseconds, and at very high hyperpolarizing potentials, it speeds up to 10 milliseconds. So I then wanted to look at the reversal potential of this channel. So this channel has to be open first. So I'm going to use this as my first stimulus to open the channel. And then instead of going back to this holding potential, we're going to go to different holding potentials. So that is the end of the first step there. And then we've switched to different holding potentials. And I've measured now the difference between the initial, right after switching, and once the channel relaxes. And I derived this graph. The reversal potential is here, where the current decreases to 0 and then is going to switch directions. That's a reversal potential. Cool. Except now, what is this channel doing? So previous work from salamander rods show that if you look at current versus voltage in the same cell, they're shaped differently, and the voltage response has this very large nose. And if you look at single photons, this is the voltage again here, it actually will peak quicker than the current response. Interesting. Oh. So you can kind of see this, but this is actually older data from Gordon.
It's just so old.
It's faded away.
It used to be white. But this is basically showing you can add cesium, which will block the IH current, and it goes from having this nose that you saw in the other slide to these very long dramatic effects that are slow. Cool, so that was salamander, that was toad. This is mouse now. So this is mouse rods, and we're looking again, the black is the current and the red is the voltage response, and it's fast, the voltage response is faster. And again, so this has all been attributed to the IH current that I've showed you. Now what about cones? So if you just kind of look, these don't look dramatically different between the current and the voltage, and if you actually normalize them, they're not much different, maybe a little bit faster, but not dramatically faster. So what is IH actually doing? Is it contributing to cones? So this is a full flash family, voltage flash family of cones, and basically I want to look at the voltage range it's going to move through. So this is actually an average of almost like 15 cones. So they're sitting at rest in the dark at minus 37-ish millivolts. They're going to peak at minus 52. Let's even say minus 55 to, pad it. Well, if we take minus 55 as the peak voltage response, and we hold the cell at minus 55, minus 60, well, IH isn't really active in the cones right then. Interesting. And further, if we look at the reversal potential that I just showed you, it's reversing at minus 55. So even if this channel is open at minus 55, reversal potential is defined as the point where current is 0. So at this peak current, IH doesn't seem to be passing a considerable amount of current where it would be shaping the voltage response. So what's going on? Let's leave that open for a second. I'm going to introduce a second current and we'll come back to it, okay? Let's talk about the synaptic calcium for a second. Now, I switched some of the solutions here to actually isolate this current, but basically I give depolarizing steps, voltage steps, and I'm going to measure in this part the peak currents elicited by those voltage shifts. So I've plotted them here. I actually did a lot more than what you can see, but the traces you just saw are these ones with the pink dots, and this is where all the business action is. So it's the important part. So this is the range at which synaptic calcium, the channels in the synapse, operate. Now these are voltage responses to sustained light, 7.5 seconds, and we're going to do a similar exercise where we run through the voltage range of these cells. So from rest to peak current, we're getting some sort of change like this, minus 35 to minus 50, minus 55, something like that. But there's also a rebound, a recovery happening. So even if we peak over here, there's going to be some sort of recovery in synaptic calcium. So we're actually looking at a range that's something like this. So the yellow is kind of rest state. The initial peak will close almost all the synaptic calcium down, and then there's going to be some sort of rebound so that the orange is going to be calcium entering during these steady state regions. Okay? Interesting. Yeah? Come on. So this is our IH reversal potential graph. This is right here, minus 55, and if we go back real quick, right, here's minus 50, minus 55 in this red region. We're about less than 10% of the dark synaptic calcium. So less than 10% of the dark calcium current at that point. Now if we get a stronger depolarization, we're going to have 0 synaptic calcium passing. You cannot encode a signal that way, right? So signals are encoded, light signals are encoded by decreasing the amount of calcium, decreasing the amount of glutamate. But when you hit 0, there's no way to further decrease calcium, and you can't encode a stronger hyperpolarization. But at these holding potentials here, IH actually is active and it's significant. So my hypothesis is that if you get a strong hyperpolarization that moves the membrane potential this direction, IH will become active and kick it back towards this range where synaptic calcium will actually work. So my hypothesis is that IH in the cones is actually helping to stabilize the membrane potential close to the dynamic range of synaptic calcium. So I'm going to move on now and show you the story of reversing the mammalian light response. So in these experiments, again I have V hold and I'm going to shift to new potential steps. And after a certain amount of time that the cell stabilizes at these new holding potentials, I'm going to flash it with light. And these experiments look something like this. So this is in the GNAT1 knockout animal again. So no rod responses. So here we go. We shift the holding potential. We give a flash of light. I've taken these flashes here, baselining them to 0 so you can see them. And then I take the amplitude of each of these, plot them against the holding potential that produced these steps. And I can plot them here. And look, it does. It reverses. So great. Super. Except, no. Okay? So if you do some math, using the concentrations of solutions, and you do some reading and look up previous measurements in fish and salamander, everything predicts that the reversal where this line crosses should be occurring right here. So this is about plus 10, and I'm getting about plus 30, plus 40. Okay. So hypothesis 1. Maybe it's those gap junctions we've seen before. We saw that signal can spread, so even though the rods are knocked out, maybe there's a cone, a lingering cone response that is spreading and confounding these results. Okay? Well, let's test it. So here is the Connexin 36 knockout. So there are no gap junctions. We do the same experiment. We isolate the photoresponses. We plot them. Again, no good. Okay. Well, all right. Maybe there's something else. Maybe there's some sort of unknown signal that's leaking, that's spreading in, and we just can't see it because there's a photoresponse on top of it. So to test that now, I add this compound, GTP gamma S. This is a non-hydrolyzable analog of GTP, and basically it gets into the molecular phototransduction pathway that you saw, and it jams it up, okay? And this causes permanent closure of the cyclic gated nucleotide channels. So in the potentials where we would see a light response, in these very negative potentials, we don't see anything, it's flat. So this would indicate that there's not signal leaking from somewhere else. There was, however, a little blip that at first I thought was an artifact and then became consistent enough that I said, no, this is not an artifact. And this little blip starts to come up. You can see this red trace. This red trace is the same trace where calcium starts to be strongly activated. So, okay, there's some strange things going on with calcium. So maybe this synaptic calcium is activated at command potentials that are critical for these reversal potential measurements. So as I'm holding the cell, I'm also activating calcium, and it's getting in the way. It's doing something. So here's where we're at. And let's look at calcium. If these are my calcium traces, which you saw, let's take the steady, or as close to steady state calcium as I can get, so this later value here. And I've plotted it now against the reversal potential, and you can see that there is a considerable amount of calcium flowing during these holding potentials. So I wanted to study the calcium dynamics in a little bit more detail, so I add this cesium internal, and that blocks some of the potassium channels so I can kind of unhide what's going on, taking some of the currents away and seeing what's left. So you can see again this red trace where it's got this really dramatic waveform happening. And that's isolated and reproduced right here in B. This is just standard solutions with cesium internal. If you add one blocker, which blocks the calcium activated chloride channels, you can see that this large this upward going bump goes away. So that's cool. And then if you add isradipine, which blocks synaptic calcium, it's pretty flat. And so you don't get this outward component because the calcium that enters through these synaptic channels will actually activate the channel that is going to mediate the bump here. So just by blocking calcium, we get rid of the entire thing, and if we add both blockers, again, it's flat. And what you can see in the IV curves now, so this black is the standard, and you can see that, through these potentials, it increased, or it got closer to 0. Okay, so now let's take that information. Let's go back to our standard internal solution. We'll block calcium, we'll block some of the potassium, and do the same experiment, isolate the flashes, and now when we plot it, we have a leftward shift. Oops. And that's exactly where predictions suggest that it should reverse. Now this condition had one more kind of plus to it, advantage, And it allowed me to isolate one last current, which is a TEA insensitive potassium current. And so I basically took those beginning traces and isolated them here, and then plotted the current voltage relationship between for this potassium channel. So the conclusions for the second part are that I saw that IH does not seem to contribute majorly to shaping the unclamped cone light response, but it actually might be working to stabilize the synaptic membrane potential close to the dynamic range of the synaptic calcium channels. I've showed you that the mammalian cone reversal potential occurs at about 13 millivolts, but that I had to block synaptic calcium in order to sort out or reveal the actual action of the reversal potential. Okay. So with that, I want to say thank you to everybody who has made it. Thank you to Sam and Gordon and the lab members who have taught me and helped me. Mike Woodruff, who originally, taught me what electrophysiology was actually about. Margie Fain produced the beautiful figures showing the molecular cascade and then channel conductances. Steve Barnes, Mark Fry, and Gabe Travis have been super crucial to moral support discussions and my experience in the PhD program. MSCIP, my PhD program, is incredible and amazing. Thank you. Jules Stein, I don't think there's really a better place to train, you guys. It's a problem. So it's been amazing. And, I could go on and on and on and thank everybody forever. So I've put a lot of initials of people who have helped me through the years and are special to me, and then of course training and funding and the people who paid for me to do this. So thank you. And then I have a few little pictures. So this is mostly the lab kind of through the years. We moved into renovated Jules Stein and painted the wall with magnet. So this is Gabby Sendek, and Kate is down there painting the wall. Alla and I at, I think, some sort of retreat conference. Ricard has been super magical in the lab, and he's incredible. It's just been really, really special, guys. And then also, besides the lab, people and, my animal friends who have really, got me through the last few years of my life. So thank you so much, everybody.
So I should explain that this is formally an examination. As Nick was quick to point out. So we will entertain questions at this point, but not from members of the committee, if you wouldn't mind. And then everybody will leave except us, and we will have some more questions for Nori undoubtedly. So if those of you in the audience have questions for her, please take this opportunity.
Katie.
So you used white light for 11 splashes, right?
No, I didn't actually. I did this really cool trick. So cones express either an M pigment, which peaks around 508, I think is the best official number, and then they also express a UV pigment, which peaks at 358. And so mouse cones typically don't express one or the other pigment. There's actually a lot of them that co-express both pigments. So instead of using white light, I'm actually using a 405 light, which is this almost really pretty purple. And this light is the isospecific point between the cone pigments, it's called. And basically it means that this light will activate both pigment types to the same degree, to the same efficiency. And that was actually a really beautiful trick because I didn't then have to sort between my cones and say, oh, this is a short wavelength cone and this is a medium, and I'm only comparing this to that. So I did at one point do a lot of analyses comparing if I shown 405 light versus one of the peak wavelengths to the cone. Does that create other downstream effects? And basically it didn't. And if I use the 405 light, monochromatic light, I'm able to activate the cones very consistently to the same degree across experiments and across genotypes. Avi?
Why do you think you still see current recovery in the GCAP recovering double knockouts?
I think they're, so even in rods, you'll see a small amount of recovery. And recovery to open channels is basically, it's all about how much cyclic GMP you have. So you have guanylyl cyclase, which is going to create the cyclic GMP, and you're going to have PD, which is going to hydrolyze the cyclic GMP, and those are balanced, or going to be balanced, and the balance between them changes depending on the light level. So why is there some recovery? Well, the dynamics of the PD and the guanylyl cyclase, the way that they balance, are such that if you turn off the PD quick enough, you're still going to be able to recover, they're just going to be able to rebound a certain amount of the cyclic GMP with the guanylyl cyclase. So the argument may be made from my data is that when you have GCAPs, you can actually accelerate the guanylyl cyclase even further and produce a larger amount of cyclic GMP to reopen those channels. Does that answer it enough? Yeah. Martha?
So these broad knockouts, do you have any, intuition for how they actually see the world? Like what kind of
the mice themselves?
Yeah.
I haven't done like behavioral tests to see like, this mouse, mice in general, when they are blind or go through a blinding mutation or have a degenerative mutation, they're not like humans. It's not hugely debilitating for a mouse to be blind, right? Like you've heard the nursery rhyme, 3 blind mice, see how they run? So they still do everything normally. I'm sure if you did put them in a behavioral test like the water maze or something, they would have deficits, especially if you were trying to do this in dim light. But as far as like their normal situation, I think they'll still be able to see in daylight. If you take them out in the room, the GNAT1 knockout animals will still be able to see things. But you could definitely find behavioral deficits if you're testing for them.
Yeah, I'm also wondering like if their circuitry adapts differently, or if there's some sort of.
It could be, and if you look at developmentally, there's definitely like activity dependent development that happens. Like there's these calcium waves that travel through the retina before there's any light stimulation. But yeah, what's happening then when you're actually getting a light stimulus, and is that affecting circuitry? I don't know, and it's very possible.
One more.
Yeah.
During in part 1,
Yes.
looking at the recovery adaptation experiment,
Mhm.
a very long light step.
Yeah.
There wasn't a stimulus bar that told me what the stimuli were.
Ah.
As during the long step,
Uh-huh.
after the
It's
first
the entire step until they turn off. Yeah.
yes, and then the short steps, then the flashes.
The flashes.
That you use to test for the adaptation. Were those all at the same intensity, or was it just time?
For the long experiment where I get flashes on top of the.
Yeah, at the very end, and they got bigger and bigger.
Okay. No. So those were increasing flashes of intensity. So when they stepped up, they were a brighter intensity. So I was trying to capture the dynamic range.
Right.
Yeah.
Okay. So what would have happened if you'd used, say, a midpoint intensity for those flashes
Mhm.
and just waited, and it was just a function of time? Is there a difference in terms of time or just intensity?
I think there probably is in these preparations. Now, they're isolated, and I don't know how much pigment regeneration is going on, so eventually you're going to have bleaching effects too. So you might, after a while, expect the cell to run down, and you would see changes in the flashes at that point, I would think. But the other thing too with these, with the whole cell I can't hold them as long as would be ideal. I can't keep the cells alive, and there's a considerable amount of rundown that happens after a few minutes.
Sure.
So that might be difficult to actually look over a long amount of time from the same cell to see what's happening. If you do the same flash over and over again, there could be rundown effects that might interact with that.
You don't see any plausible mechanism that just waited time in that shorter period, it was 10 seconds or something after that, that time itself would play a role in the recovery?
I would expect it to, but the cones also recover so fast that I think that you're seeing most of the recovery already. I think that especially the entire trace was 40 seconds of the background. And it gets pretty stable. It's not like you see this huge gradual or noticeably gradual decline much longer after that. So I'm not ruling out that there is a longer adaptation that could occur and will affect the flashes, but I do think the majority of it is happening within those first several seconds.
Great.
Yeah.
Okay, if there are no more questions, then I think we'll take our let everyone
I know. How you doing?
Hey, Lauren. This is your thing.
Thank you. Thank you. Thanks, Annie. Thank you. Thank you. Thank you. Thanks. If you guys wanna stick around for an hour or so, I can find some
It
out.
won't even be that. Yeah, like half an hour, 40 minutes.
This is Gordon. Did you get to meet Gordon?
Hello. I've heard so much about
actually, if you only find Margie, she could probably put you someplace.
Oh,
Or at least take you up to the lab or something.
Yeah.
You have my desk or something.
like that? Okay. Margie.
Yeah. She's just around the corner. We left the lab open.
Okay.
Alright, it's good
Yeah.
to meet you.
Nice to meet you.
Tell Margie how much you like her turkey.
Margie's your wife. Alright, you were great.
Thank you. I just